Hello, everyone, and welcome to today's webinar on Phase 1 dose escalation trials with Escalate. I would now like to introduce Pantali Swahos, Principal Strategic Consultant at Cytel.
Thank you, Alyssa. And once again, thank you, everybody, for attending this webinar, the second one on a series of East orientation webinars. Today's topic, as you see on the screen, is going to be Phase 1 dose escalation trials applied with Escalate. I will guide you through the methodology that is available in our system. I will only try to scratch the surface and mention some of the options because this is a topic that can be covered in probably 2 or 3 hours. And as last week, I will delve into a case study so that we can see the various options of the software, both in terms of simulation as well as when you're executing your dose escalation study. I will be more than happy to field any questions that you have. And in the end, we're going to go to the conclusion of today's webinar. So with that, let me start with a brief introduction of the topics that we're going to be covering today. As you probably already know, EAST is modular, so there are different modules that will allow you to deal with different types of trials for different types of endpoints, different phases. And today we're going to be picking up 2 of these modules. The first one is the Escalate module, which allows us to design a dose escalation study geared towards identifying a maximum tolerated dose. But I'm also going to show you a little bit of the program module, which is the module that we're using for designing a sequence of studies, which is going to be handy in today's case study, as you will see in a little while. So Escalate is our adaptive Phase 1 dose escalation module. It contains a variety of methods, both for single as well as a combination of agent designs. It aims at characterizing in an optimal way the maximum tolerated dose and serve as a clear communication tool with your internal clinical team through the tabular and the graphical displays that it produces. That will allow you to improve screening and selection of the active agents to carry forward onto your study. On the other hand, the second module that I'm going to be illustrating today as well, which is the program module, allows you to simulate a sequence of studies. All of the modules that I showed you in the previous screen of these allow the simulation of a particular clinical study many times. So you can simulate 10,000 replications of a particular clinical study, and in the end, calculate the power of the study, probability of success, etc. With the program module, you're allowed to simulate a sequence of studies. So in this case, we will have a dose escalation study. And once the maximum tolerated dose is identified, we're going to be following that with a cohort expansion study. And then for the sequence of the 2 studies, we're going to be able to get an idea about the overall chances of success of the study. The program module also has other applications, as you will see as you can see on the screen, especially taken in the area of oncology, where you can follow up any single arm study with a group sequential, study using the log-rank test statistic. But today, we're just going to be focusing on the dose escalation part, followed by the cohort expansion part. In terms of the methodology, let's set a little bit the terminology that we're going to be using. What we want to do is we want to assess the dose toxicity relationship and see how the different dose levels affect the probability of a dose-limiting toxicity. Our observable quantity is going to be those dose-limiting toxicities, right? So you're either going to have a dose-limiting toxicity or not. So naturally, there's going to be a binomial type of distribution that's going to be modeling this dose-limiting toxicity. Our goal is to determine the maximum tolerated dose or the recommended Phase 2 dose, RP2D. And this is the same either in a single agent setup or a combination of agents setup. In the latter case, you might explore different methods for modeling also the synergy between the 2 agents. So you can have an interaction term that essentially will govern whether the two agents are synergistic, antagonistic, or whether there is no interaction between them at all. So going back to the setup, we are assuming that we have k many dose levels, number d1 up to pk. Each one of them has an unknown probability of toxicity p1 up to pk. The higher the dose level, the higher the probability of toxicity. This is the basic assumption that we're making. Our universal goal is to find the maximum tolerated dose, which is defined as the highest dose level that will have a toxicity rate which is close to but lower than a fixed rate. For oncology type of studies, this rate ranges typically from 20 to 35%. 30% is a rate that's universally being used, especially when people are using one of the most prevalent methods in the field, which is the 3+3 design. The collection of designs that we have available in EAST range from the most prevalent one that I just mentioned, the 3+3 design, which is a classical algorithmic type of an approach targeting either a 33 or a 17% rate of toxicity. We have the CRM and the BLRM, continual reassessment method, and the Bayesian logistic regression model, which are model-based approaches. They're Bayesian in nature, so they utilize the Bayesian updating, and they provide a model for the dose toxicity relationship, which is going to be updated every time you get another cohort of subjects. And we also have a hybrid between these two, the modified toxicity probability interval approach, which has some of the advantages of both methods in the sense that it is very easy to use, much like the 3+3 design, but it also has some Bayesian aspects into it. The double agent method that we have available is an extension of the Bayesian logistic regression model. And we also have the PIPE method, which was developed at MRC at Cambridge, which is a method for identifying not a maximum tolerated dose, but a maximum tolerated contour in the two-dimensional space of your agents and probabilities and their corresponding probabilities of toxicity. Just a quick run through the methods. The 3+3 design, as I said, is the most prevalent one. It has been used by over 90% of the studies that are geared towards finding a maximum tolerated dose. There are different flavors of this design, depending on how conservative or not conservative the method is. Typically, the degrees of conservativeness have to deal with how the method approaches situations when you have one toxicity out of the last 6 subjects, or whenever you have 2 toxicities out of the last 6 subjects. Within EAST, we are using 3 flavors of this method. What we call the 3+3 low, which is the most conservative, the 3+3 high, which is the least conservative, and sort of like a middle choice between the two. Apart from that, we have the continual reassessment methods. Again, the objective here is to find the dose that yields a desired toxicity level. And you do that by performing a succession of interim analyses. And at each interim analysis, you fit a parametric model to the data. Typically, it's a 1-parameter model, whether it's a 1-parameter logistic, a power function, or a hyperbolic tangent. And with that, you estimate the probability of response at each dose level. This estimation of the probability of response is done in a Bayesian fashion. And then there is the interim analysis decision part, where you allocate the subject in the next cohort to the dose level that has a posterior probability which is closest to the target that you have set in the beginning. The third method differs from the CRM in the sense that, first of all, it is utilizing a 2-parameter logistic function. And instead of targeting a single threshold, a single desired toxicity level, it targets an entire interval of desired toxicity levels. So we're going to be getting in a little bit more detail of this in this method, since this is the one that we're going to be illustrating. So the 2-parameter logistic model that's being used is the one that you see in your screen. The 2 unknown parameters are the slope and the intercept term of this straight line, the logic scale. The authors of the original paper, Neuschwander et al., introduced also a reference dose, which allows you to calibrate the values of these parameters and to view the intercept term as the odds of the dose-limiting toxicity at that dose level, at that reference dose, while the slope term is viewed as an increase in the log odds of a DLT for a unit increase in the log dose. So this is the model, and the way that it works is by, first of all, assigning a prior distribution to these parameters of choice. So these two parameters, log alpha and log beta, will have a bivariate normal distribution, which is not that easy to decide what that distribution is, but we will see how we help you determine that distribution. Then the next cohort of subjects is going to be assigned to the dose level that either minimizes the posterior expected loss or the Bayes risk, therefore undertaking a pure Bayesian approach besides the likelihoods, which is formed from the logistic function, the prior distribution, which is formed from this bivariate normal distribution. You also impose a loss structure depending on whether or not you fall into this targeted toxicity interval. Once you have these three elements, then you can naturally construct the posterior expected loss and therefore assign the next cohort in the dose level that minimizes that. However, there are several subjective quantities in this type of an approach. Besides the prior distribution, you also have to determine the loss structure, and therefore there is another alternative for this approach, which is the escalation with overdose control. This is a constraint optimization approach, which essentially fixes the probability of overtoxing or overdosing a subject at a cap of, say, 25%, as you might see in this example here. And then based on that constraint, you're trying to maximize the chance that you fall into this targeted toxicity interval. Again, a posteriori, through the use of the posterior distribution. So this is the approach that's being used. Some of the details that come in the application of this approach are going to become apparent with the case study that we're going to be revisiting. So the case study is actually one that was published in the New England Journal of Medicine on non-small cell lung cancer. It's a paper by Shaw et al. 2014. And yes, you can see is a phase 1 multi-center open-label dose escalation study where the agent was administered orally in adult patients with tumors characterized by genetic abnormalities in anaplastic lymphoma kinases. So in terms of the parameters that we're going to be entering into our software, there's going to be the maximum number of subjects that we're going to be considering, which in this case is going to be 40. I'm just going to increase that just for by a little bit to 42 in our software, just to show that initially we can be dealing with cohorts of size 3. So we're going to be exploring this maximum sample size. Of course, you may reach that maximum sample size or you may stop earlier due to some stopping criteria, which we will see later on. The module that we're going to be utilizing is the one that I showed you exactly. Again, these screenshots are taken from the paper, and which is the logistic regression that I mentioned before, where we have a reference dose of the 350 milligrams of the agent, and the target is about 33%. As we said, the target is actually going to be defined in terms of the intervals of toxicity. We're going to have an interval of underdosing when the probability of toxicity is between 0 and 16%, an interval of targeted toxicity when the probability of toxicity is between 16 and 33%, and anything above that is going to be considered excessive toxicity. We're going to be utilizing, again, the escalation with overdose control principle, which means that we're going to be capping the probability of overdosing at 25%, and we're going to try to maximize the chance that we fall into this targeted toxicity interval. A few more pieces of information about the study. We have 9 dose levels that we are examining, from 25 up to 1250 milligrams. 50 milligrams is going to be our starting dose. the way that the software works is through simulation. So basically what you have to do is you have to see how well the method performs in identifying the MTD in situations where you know what the actual MTD is. So these situations that you see here, these 5 scenarios that were listed into the paper, are situations where the true MTD appears at different places in the range of the dose levels. You can see in scenario 1, it is somewhere between 100 and 200 milligrams. In scenario B, is between 600 and 800 milligrams. In scenario C, is between 1,000 and 1,250 milligrams. And the way, what we also try to make apparent in these types of scenario is the way that we reach that MTD, whether we do that with a steep curve or whether we do that with essentially a flat curve. So there are different approaches, different scenarios that we will consider. And then we will see through the power of simulation how well this method is going to be doing in picking up the true MTD. There is also the execution mode of the software, which I'm going to be illustrating as well, which is what is actually going to happen when you start recruiting subjects and applying this methodology. I mentioned earlier that our maximum sample size is going to be of 42, but the method, if you just leave the criteria that I mentioned earlier in terms of how the escalation is done, has the potential of essentially skipping some dose levels, which may not be seen as safe, especially from clinical teams. So you can impose different restrictions in that. The type of restriction that was imposed in this particular paper was that the dose increment should not exceed 100% of the current dose. So you, at the worst case scenario, you're going to be doubling your current dose level. And then we have to decide what sort of prior parameters we're going to be using, prior distribution we're going to be using for these 2 parameters. So you can see the bivariate normal distribution that was used by the authors. You can see it up here on the top. Through the use of the software, we're going to be able to visualize that prior distribution in the plot that you see down here, which not only gives you an idea of what is the median of the probability of a DLT at each dose level, but also what these standard deviations result to in terms of uncertainty, of prior uncertainty. And this is, in this particular case, the prior uncertainty that was undertaken in this particular case. So you are almost certainly assuming that there's going to be a flat prior at each dose level. So you're not trying to bias your results by imposing too informative priors. Finally, there's going to be a stopping rule that we have to think about because the way that this works is that you may either let the study reach its conclusion by exhausting the number of subjects, the maximum number of subjects that you have, which in this case is 40, or you might stop if different criteria are being met. Within EAST, we entertain different types of criteria, which include some of the ones that the authors ended up using. So the authors ended up using a criteria that's called the target rule, which stops the trial if you reach the targeted toxicity interval at least 60% of the time. Remember, our goal is to maximize the chance that you fall into that interval. So if you end up in that interval more than 60% of the time and you have expended at least 6 subjects in that dose level, then you should be stopping there. There's also an allocation rule that was applied in this particular study, which if you have expended at least 2 cohorts of subjects in the current MTD, then you would also stop the study. And once you select these stopping rules, they're going to be populating this table on the right of the software and will allow you also to prioritize these rules. So this is the setup that we have. Let's go into EAST to get an idea about how this works. So you would select under the dose escalation module the method that you want to select, which is the Bayesian logistic regression model, and you will be shown 4 tabs, which are the standard simulation tabs that we have available into the software. In the first tab, you would specify what your target probability of toxicity is, if you have a single number. But also remember, these are the intervals that were also specified into the paper. So the targeted toxicity interval is anywhere between 16 and 33%. The reference dose that was used was 350 mg, and the probability of overdosing was capped at 25%. Now, the way that we can, of course, go ahead and fill in the prior values that the authors put in. But what we're also doing in the software is we're making it easy for both the statisticians and the clinicians to have an idea about what this prior distribution means. Because if you view it plainly as a prior distribution on log alpha and log beta, this is not going to tell you much about sort of like the chances of a probability of DLT at any dose level. Instead, what we offer is this prior calculator, which allows you to elicit from your clinical team essentially 2 points. Because in the logit scale, this model that we're using is a straight line. So if you can determine 2 points on that straight line, then you can essentially determine the entire line. So you can elicit from your clinical team either the chance of a toxicity of the lowest dose level and your best guess at the MTD, which could be done in this form, and then you would recalculate and see what that means in terms of your prior distribution, or you can elicit the chance of a probability of toxicity of a lower dose and the highest dose, or the chance of probability of toxicity of the lowest dose and the reference dose. This is utilizing a methodology that was developed by statisticians at Roche in Welland Garden City, Hassan and Kinnersley, which enables us to make this translation from this probability space of the dose-limiting toxicities into the space of the prior distribution for log alpha and log beta. The reference is going to be available in the slides that we're also going to be sharing with you. So this is the part that we're going to be adding. This is how the prior distribution is going to be determined. We are also going to be determining how we're going to be skipping dose levels, if we're going to be skipping dose levels. And following what the authors did, we're only going to be allowing at most a doubling of the current dose level with clicking OK. Then we have the different stopping rules that will be allowed. And as we mentioned, we're going to be using the target rule, which will stop the study if we reach the targeted toxicity interval more than 60% of the time, as well as the allocation rule. There is one more tab which allows you to set the different response generation scenario. So this is a case where you would actually say, well, I do have these 5 scenarios that I wanted to put in. One thing that you can do is you can actually copy all of them from your Excel spreadsheet, and then you can paste them right into this area so that you can determine the 5 different reality scenarios that you're going to be using. So you can either enter those manually, or you can create scenario from different parametric families as the ones that you see in this drop-down menu. Finally, this trial is going to be simulated 10,000 times, and we're going to be counting in each one of those 10,000 applications how often we detect the right MTD. So what I'm going to be showing you here are the actual outputs from the 5 different scenarios that we run here. This is initially the summarized output. So the summarized output here shows you that we have scenario A, B, C, up to E, as you can see here. And in blue and white, you will see a repetition of the inputs. In yellow, you will have what things that were actually calculated in the design. So, for example, here you can see on scenario 1, this was a scenario where the true MTD was actually 200. And then you will see what the actual procedure picked as the MTD, which is the dose level that was picked the most often, or the mode from this method. And you can see that it underestimated the MTD for this reality scenario. when the reality was a dose level of 800, the true MTD was 800, it actually picked that correctly. It did the same thing with the true MTD was 1250. It picked the correct MTD. In the other two cases, when the MTD was 600, it underestimated it by selecting 400. When the MTD was 1,000, it underestimated that by picking 800. Okay, so the way that you would select and you see what this option does is by essentially viewing this summary table, and that would be sort of like a very quick view that allows you to see how well the method did in picking up the right MTD. So it might seem that it did not do very well. It actually does much better than some other designs. Like, for example, if you compare this to the 3+3 design, which I can show you right here for the 5 scenario that we selected, you will see that the 3+3 design consistently underestimates the MTD. MTD was 200, it selected 200. Was 1,000, it selected 800. Was 600, it selected 400, and so forth like that. Okay, so, and this is a phenomenon that has been identified quite often with the 3+3 design. So the design can then, after you select this design, you can go and execute your study. So when you're executing your study, you will select the so-called interim monitoring dashboard, which will tell you, well, enter the data that you have. So you can start, if you start at the 50 milligram dose level, you can enter the number of subjects that you have and how many DLTs you observed, and you can click on OK, and you can see what the next recommended dose level is going to be. Remember here we had put in the restriction that at most we can double the dose level, and you can see that the next recommended dose level is 100. How do we arrive at this decision? Well, all we have to do is we have to see what are the chances of falling in each one of the 3 intervals. The blue interval, which is the underdosing for each dose level, the green interval, which is the interval of proper or targeted toxicity, and the red interval, which is the interval of overtoxicity. So if I turn off the underdosing and targeted toxicity intervals for the moment, what you first can see is the chances of overdosing at each one of the current dose levels. And you can identify that from dose level 400 and downwards, all the dose levels have a chance of toxicity which is below 20%. Remember, our cap was 25%. So this is the set of admissible dose levels that you would have in your actions. Then among these admissible dose levels, you need to pick the one that has the highest chance of falling into the target toxicity interval. So if I were not using any constraint in my BLRM, dose level 400 would be picked as the one that has the highest chance of falling in the targeted toxicity interval while maintaining the constraint of not being above 25%. And of course, this is skipping essentially 3 dose levels from the dose level that we began with. Of course, here we are using this constraint, so the actual dose level that was picked was 100. But you can see through this display that we're making available to your team how high you could actually go based on the criteria that you had set up. Another very useful piece of information that you have in here is the predictive distribution of the number of dose-limiting toxicity. This will show you for the next dose level, as well as for any dose level in the future or in the past, in a cohort of size 3, what are going to be your chances of having 0, 1, 2, or 3 toxicities. So for the next dose level of 100 milligrams, you can see that you have close to a 90% chance that you're going to have no toxicities out of a cohort of size 3. You're going to have a less than 10% chance that you're going to have 1 toxicity out of 3, and a less than probably 3% chance that you're going to have 2 toxicities out of 3. So the odds are pretty good. And then you can also check this for any other dose level ahead of you. So if we go to this 400 dose level that the unconstrained BLRM will give you, you can see that still the chances of no toxicities out of 3 are pretty good. They're close to 70%, close to 20% of having one toxicity, less than 10% of having two toxicities, less than 5% of having three toxicities out of three. So that's why we say that this tool serves in real time as a communication tool for your clinical team, because as a statistician, you can create these types of displays on the spot, and you can bring these to the table and see what the decision should be for the next cohort of subjects. Finally, what I would like to mention is the cohort expansion capabilities of the software. Once we identify the maximum tolerated dose, we can design a single proportion go-no-go rule study, a single arm cohort expansion study, and you can determine, you can enter as inputs into that study what sort of sample sizes you're going to be considering. In this case, a sample of size 10. What is going to be a clinically meaningful level? In this case, 10%. What would be the desired level of clinical activity? 20%. So this corresponds essentially, for those of you who are familiar with the go-no-go terminology of Lalonde et al. Or of Fruer et al. At AstraZeneca, it corresponds to the lower reference value or the target value in your go-no-go decisions. And we're going to be undertaking a Bayesian approach as well by utilizing a beta 1-1 or uniform probability distribution for the chance of success. And the scenario that we're going to be considering here is that the true response rate is going to be 0.1 for any dose level below the true MTD and 0.2 for all dose levels at the true MTD or higher. So going back into EAST, what you would have to do is you would have to click on the program simulation icon and select the dose escalation with cohort expansion. What you will be shown at that point would be this graphical panel where you would be allowed to select what method you're going to be using for your dose escalation study. Here I used the BLRM, and I actually used the exact same BLRM that was that appeared on the Shaw et al. Paper with the same rules, as you can see. And then following that, you can add the next study, which could be either a single mean go-no-go rule, a single proportion go-no-go rule, or a single survival go-no-go rule. So the one that we selected here, which I had on the slides, was a single proportion go-no-go rule. And you can see here we are considering a maximum sample size of 30. And we have our go target to be at 0.1, our no-go target to be at 0.2, the low reference value and the target value. And you want to have an acceptable risk of 80% of meeting the go target, and desired confidence, of meeting 80% of meeting the first target, and an acceptable risk of 10% of meeting the second target. And you can see actually here how your go-no-go path looks like. And for those of you who are familiar with these type of methodology, what you're trying to avoid is the gray area or the consider zone, the chance of falling anywhere between a go and a no-go decision. You do not want the chance of doing that to be high. So once you have selected the single proportion study that you have, then you can simulate the sequence of two studies. So this is going to be running 10,000 replications of this sequence of two studies. And in the output, you can identify first anything that is related to your dose escalation. You can see that your true MTD was 800. And this is exactly what the BLRM ended up picking. It picked the correct value about 36% of the time. And the next one, the next highest percentage was on the dose level 600, which was also within that targeted toxicity interval that we considered earlier. So this is the part of the dose escalation part. And as you scroll down, you will get also the cohort expansion part, which tells you what was the average go cutoff, the average no-go cutoff, and what percentage of times with these, after you had determined the MTD, you ended up with a go decision, a no-go decision, or a considered decision. So you can see here that the considered decision was slightly above 30%, about 32%. One of the golden rules that Lalonde et al. Or Fruer et al. Had put in is that you typically want this probability to be below 30%. You don't want the process to be too ambiguous. So this leaves room for further tuning, again through simulation of the success criteria to allow you to design a more efficient cohort expansion study. So in the slides that follow, I have gone step by step through what I just described in the demo that I mentioned. And I'm going to be following that with the references for the methods that are available in EAST. And with that, I would like to thank you for attending today's webinar. And I would like to open up the floor for any questions that you might have.
Thank you, Pantelis. We are now going to begin answering questions submitted during today's presentation. So Pantelis, our first question is, does EAST design a trial with simultaneous efficacy and toxicity?
Currently not. I think, well, the only version of simultaneous efficacy and toxicity is actually what you just saw here when you have the toxicity part governed by your dose escalation study, and then you have the efficacy part governed by your dose expansion study. We are aware, of course, of methods that simultaneously do that. There is the FTOX method by Thor et al. There is the TEPI approach, toxicity and efficacy probability intervals approach by Lee and his team. And we are exploring these methods so that we can add these functionalities further into EAST. But currently, the only version that you will see in EAST of treating efficacy and toxicity at the same time is through the sequence of dose escalation followed by a cohort expansion.
Thank you, Pantelis. So we have another question. Do you have any recommendations on how to set your priors?
Okay, so this is a very tricky part. So setting the priors is not an easy task. And of course, we do not have time in this 30-minute webinar to go into the details of this. Obviously, what we offer in EAST is an easy way to elicit such information from clinicians, and essentially, after viewing the resulting prior, see how you can fine-tune that, maybe make inflating a little bit the variance, making it as non-informative as possible. There are, however, some methods that have been used, especially in the following up the development of the BLRM by the authors of the original paper. And there is a meta-analytic prior predictive method, which allows you to essentially run a meta-analysis on historical studies and use those to form essentially a prior distribution. So you're going to be creating essentially a normal distribution, which is going to be a result, which is going to be essentially a mixture of other normals, and to alleviate again the chance of inflating your false positives. What you want to do is you want to add a robustification component into that mixture. So this map prior approach from Neuschwanderer, Smedley et al. Is one way to deal with this. And then there are also other approaches in the literature that have been made available lately in how to essentially borrow information from historical studies and translate that into a prior distribution that you can use. There's nothing automated done currently in EAST. We are developing a product, an extension of EAST, which allows us to use some of these methods for Bayesian borrowing, and then embed the resulting prior into the dose escalation procedure.
Great, thank you, Pantelis. I think we have time for just one more question here. Do you plan to implement MTPI2 soon?
MTPI-2 is actually already implemented. I did not have a chance to go through MTPI at all. One of the criticisms, well, MTPI, as I mentioned at the beginning, is a very nice procedure because it offers ease to the clinician, and it has some of the attractive quantities of a Bayesian model by essentially imposing a beta distribution on the probability of toxicity at each dose level. The current or the original implementation of the MTPI suffers from the Occam razors problem. If you view, for example, the resulting trial monitoring table, you would get into situations where you might have a cohort of subjects of size 6 and 3 toxicities, so about 50% to be toxicities, and then there is the action that the program would recommend would be to stay at the current dose level when you would probably have to de-escalate. So this has been fixed in MTPI-2, and we have implemented it in version 6.5 of least.
Great, thank you. And if there are any questions that we were unable to get to, we'll be sure to forward those on to Pantelis after the webinar is complete today.
Thank you, Lisa. So, first of all, what I would like to mention for those of you who are attending, so we have, oops, I have to go back a little bit. This is the second webinar of the series of 5 that we have already attended. Last week we saw the MAMS module for designing adaptive umbrella trials, and what's coming up next week, we're going into phase 2 now, into adaptive dose finding, and we're going to be using MCP mod methodology for that. And then we have some confirmatory adaptive designs coming in 2 and 3 weeks with the use of unblinded sample size re-estimation as well as adaptive population enrichment.
And I just want to take a moment to remind you that EAST gives you access to a wide variety of trusted fixed and adaptive trial designs. EAST is used by nearly every major pharmaceutical company and the FDA. With a broad selection of popular designs in an easy-to-use format, we can help you quickly create and compare trial designs. Our company also offers a range of services from staff that act as an extension of your team and consulting services for more cutting-edge projects to our new real-world analytics capabilities. We are here to help support your objectives.
Thank you, Lisa. And once again, thanks to everybody for attending today's webinar. I will be more than happy to entertain offline if you have any questions. You have my contact information, and I'll be more than happy to answer any questions you have and help you out with your trial. Thank you again.
Thank you, Pantelis. On behalf of Cytel, thank you for joining us today and have a great rest of your day.